Hi everyone, and welcome to this lecture on Chapter 7. So in Chapter 7, we start to talk about metabolism. We've mentioned this concept a little bit so far. Certainly I've probably said the word metabolism in the past 3 lectures when we've talked about carbohydrates, fats, and proteins. So this content is still in Unit 2 when we're learning about the macronutrients. And the metabolism, as you'll see, is basically the way that we process our macronutrients. So metabolism we can define as the sum of all chemical and physical processes by which the body breaks down and builds up molecules. So this is an important piece right here, and builds up. So we're talking about both processes that break things down, right, which is basically digestion. It's not exclusively digestion, but of the things we've learned so far, digestion is this process of breaking down our foods into these smaller components, right? We've talked about breaking carbohydrates, breaking these long polysaccharide chains down into each individual glucose molecule. We've talked about breaking triglycerides down into the glycerol backbone and the 3 fatty acids. And we've talked about breaking these large, very complex protein molecules down into their component amino acid parts. So this process of breaking things down is one aspect of metabolism. And then the process of building new molecules is another aspect of metabolism. So especially in the protein lecture, I talked a lot about how when we eat protein from our food, if it comes from an animal or it comes from a plant, we don't want the other animals' proteins, and we don't want, the black bean proteins. We want to make our own human proteins. And so we break those proteins down to get the amino acids, and then we use those amino acids to build our own protein molecules. Going back to the carbohydrate lecture too, when we talked about blood glucose regulation, we talked about the processes of gluconeogenesis. Remember, that's the process of making glucose molecules from non-glucose substrate. We actually typically do that from amino acids as well. And we can also make glucose from stored triglyceride. So that would also be an example of building up new molecules. And then going back to breaking down, we also talked about glycogenolysis, right? Glycogenolysis, that crazy complex word. But remember, just break the word down as I'm saying it. Glycogenolysis, right? Glycogenolysis, breaking down glycogen. And remember, glycogen is a way that we store glucose in our liver and in our muscle cells. And so when our blood glucose levels are lower than normal range, we may, if we haven't eaten a meal to get new glucose in, we will break down our stored glycogen to provide glucose to be available to raise blood glucose levels. So all of these, so much of what we've been talking about so far is effectively metabolism. We just haven't quite called it that. So that's a long introduction, but really, really critical that you understand what metabolism is. So within nutrition, we can well, specifically, I guess when we're talking about breaking down as part of digestion, we're also exploring the calorie content of our food. So we do have some ways of measuring, I guess, the metabolic effect of foods. So we have something called a calorimeter, which is basically a way of determining how many calories are in a food. And remember, calorie is a unit of energy. So by determining the amount of energy in the food, we determine the amount of calories in the food. And when we're breaking our food down, we're also going to get into we're also going to get into the next phases of metabolism, which are further breaking down our building blocks. So we're going to talk about how we break glucose down to release energy, right? We've said that glucose is the primary energy substrate in the body. We've said that triglycerides are also an important source of energy. So this next phase of metabolism that we're going to be talking about is going to be looking at actually breaking down these building blocks of our food, these building blocks of our macromolecules, so these individual nutrients. So when we break glucose down further, when we break triglyceride down further, when we break amino acids down further, we do that specifically to provide calories to the body, to provide energy to the body. So this idea of being able to measure the amount of calories in a food it is important because it gives us a sense of how many calories then, how much energy then we're taking into the body. So chemical reactions in the body require or release energy. They either require or they release energy. And sometimes, honestly, they do a little bit of both. So what we're going to see is this process of breaking down. This process of breaking down will tend to release energy, tend to release calories, make calories available to the body. Whereas this process of building tends to use energy, right? And that should make sense, right? If you're going to, say you're like, I don't know, remodeling a home or something, or you're building a home. If you're building a home, you need to put quite a lot of energy into it. But say you're going to do, I don't know, say you have a brick wall, you built up a brick wall, but suddenly you decide you don't want it. If you take the brick wall down carefully, you suddenly have all these bricks then available that you could use for some other purpose, maybe building a sidewalk or something, or a little fire pit or something like that. So generally speaking, when we build up new molecules, we put energy into that process. And when we break down molecules, we tend to be releasing energy, making it available to our cells. So cells require chemical energy to grow, reproduce, repair, and maintain body function. So our bodies are basically in a constant state of metabolism. So any of these things, growth, reproduction, repair, and just general maintenance, we can call all of that metabolism. Okie dokie, sorry, I think I spent a little too long on that first slide, forgive me. So there are two parts to metabolism that you'll learn about for this class. And then again, if you take physiology or anatomy or any other biology class, you'll probably revisit this concept of metabolism in other forms. So we have, let me actually go back one more thing, I'll write it out here. So we have two aspects of metabolism. Metabolism equals catabolism. Let's see if I can write this with my finger. Catabolism. And we have this process of anabolism. So we can say that metabolism is effectively the sum of all the processes of catabolism plus the sum of all the processes of anabolism. So let's talk about anabolism here. Alrighty, so anabolism is the process of making larger, chemically more complex molecules from smaller ones. So what is that? Is that building or is that breaking down? Making larger, complex molecules from smaller ones. That's building. So anabolism is building. So write that down on your notes. Right, anabolism is building. So when do we see anabolic processes? Anabolic processes are processes of anabolism. We see anabolic processes during growth. That makes sense, right? We're building new tissue, building new cells. Repair is also similar to growth, right? But if there was damage or if there was illness or injury, we need to repair that. Again, just maintaining normal function of the body. And synthesis of chemical products essential for human functioning, right? Kind of goes in line with maintenance. So a lot of what our bodies do throughout the day are in these processes of growth, repair, and maintenance. And again, anything that is building, generally speaking, and at least for our purposes, we're going to say requires energy, right? Again, anytime we build something, we need energy. So again, going back here, when we say breaks down and builds up, I could say breaks down is catabolism and builds up is anabolism, right? And I'll tell you my fun way for remembering catabolism. I have 3 cats. I'm a proud cat lady, or sometimes, I guess. And so if anyone has experienced cats before, you might recognize that they can be a bit destructive, right? They tend to break things down. Like you can't have nice furniture because it's going to get clawed to shreds. So this is my way of remembering which is which. Catabolism, I always think of my cats, and I think of, they're a little destructive. So they break things down. They break down larger, well, cats don't, but, well, they do, but in our bodies, right, we have these catabolic processes, and these catabolic processes break larger complex molecules down into smaller ones. So again, the processes of digestion, breaking those large proteins, breaking the large lipids, breaking those large polysaccharides down into their component parts, meaning their monomers or their building blocks. So breaking the protein down to the amino acids, breaking triglyceride down to glycerol and 3 fatty acids, breaking carbohydrates down to glucose, or maybe there's fructose in there, or maybe there's lactose in there, or galactose, excuse me. And so, yeah, so these are the 2 names for breaking down and building up: catabolism and anabolism. So please do remember those. Other times that we see catabolism in the body is when, again, actually the beginning processes of growth and repair. Oftentimes to grow, we first break down old cells or old tissue so that we have space and even some resources to rebuild new tissue. And so again, the processes of catabolism for our purposes, please remember, these are going to release energy, release calories, making calories available to the body. So going back here, for some reason my slides get a little out of order, and I noticed some of them when I prepped the lecture, but clearly I don't notice all of them. So this, again, I took out the figure number from your book, but it doesn't matter if you're using different editions, it wouldn't be the same figure number. So look for this in your chapter 7, it would be early on. But this hopefully is just a nice chart to help you have a visual look at anabolic and catabolic processes. And so you'll see they're not separate from each other, right? Anabolic processes can feed right into catabolic processes. So again, an anabolic reaction might look like building glycogen. So glucose, right, in the liver especially, we would, and in the muscle tissue, we would join all these glucose molecules together to build this large chain of glycogen to store the glucose. Same thing with glycerol and fatty acids. If we have a big excess of glycerol and fatty acids, we're going to build those up into little triglycerides and store them in our adipose tissue. And then the same thing, anytime, all the time, we're building body proteins. So we take the amino acids from our food, from the amino acid pools, and we join amino acids together to build protein molecules. So building processes are anabolic, right? Again, if you need to remind yourself, right, building. And then on the other side, we see the breakdown. So we see that catabolic processes take the large complex molecule and break it down into its component parts, right? So glycogen gets broken down into glucose, and this releases energy. I did point out over here in the anabolic reactions, you'll see the energy and the arrow going into the reaction, the arrow going in, the arrow going in, building requires energy. So that would be another thing to jot down, right? Requires, or you could say uses energy, and remember energy equals calories. Right? And then on the right side, all of these catabolic processes, triglyceride being broken down into its component parts, and then breaking the protein down into its component amino acids. All of that releases energy, right? So catabolism, if you like my cat analogy, please use it. But catabolism is breaking down and it releases energy. So hopefully it just takes a couple seconds to kind of commit your brain to some form of memorizing this. And then you're set for life. You'll always remember catabolism and anabolism. Alrighty, so what we're going to introduce, maybe for the first time in this lecture, is this concept of adenosine triphosphate, or ATP. So when I keep saying over here that we release energy or that we use energy, that the most basic form of that energy, I guess we could say, is this molecule, adenosine triphosphate. So adenosine triphosphate, abbreviated ATP, is a compound used by cells as a source of metabolic energy. So in previous versions of this text and other nutrition textbooks that I've used, ATP is considered the energy currency, which is a concept I think I kind of like. I don't know that it's written here, but it's the energy currency of the cell, right, or of the body. So when we need to put energy into building new protein molecules or building glycogen or building adipose tissue, we need to basically spend, if you want to think about it that way, ATP. We need to use ATP in order to make anabolic processes go. And in the same but opposite direction, whenever we're breaking down our larger molecules, so when we break down triglycerides, or we break down glycogen, or we break down protein, we're releasing. It's like you break your piggy bank apart and suddenly you have all these coins, all this cash, all this currency. So when we break things down, we release a bunch of ATP, we release a bunch of energy currency, if you will. So we're going to take a look at ATP as a molecule on the next slide, and you'll see that there are these bonds between the phosphate groups. And so let me hop back up here to the name of the molecule. It says triphosphate. Again, just take a hot second and think about what that means, right? Tri means 3, and phosphate means we're talking about 3 phosphate groups, 3 phosphate compounds, or potentially even just 3 phosphate atoms. So we'll look at that on the next page, but basically we have these 3 phosphate molecules or atoms bound to each other. So 3P, we could write that out like this: phosphate bound to phosphate bound to phosphate. And it's in these phosphate-phosphate bonds that we have all this energy. So that's why ATP is this energy currency, is because of these phosphate to phosphate bonds. So when the bonds are broken, energy is released. And similarly, we do make ATP all day long. So when we join these bonds back together, we're technically storing energy. It's like putting coins back into your piggy bank to make them available for some later use. So the energy is used to keep cells functioning, and then a small amount of ATP, like I sort of just said, is stored in every cell, generally for immediate use. So I guess I didn't say that. I thought I was going to say something else. So at any given time, your cells contain a small amount of ATP. And then again, when we eat food, we're releasing a lot of ATP from the food, which we can then use to replenish these little ATP stores. So our bodies both break down ATP and regenerate ATP. We're going to talk about that in this lecture. Okay, so here is, let's look at this top part. And here is ATP. So adenosine, kind of like an amino acid backbone, actually. Something like an amino acid, I guess. It's not exactly an amino acid. But so you have adenosine and then triphosphate, so 3 phosphate groups. So here's 1 phosphate, here's a 2nd phosphate group, right? And here's a 3rd phosphate group. And so you'll see these high energy bonds between the phosphate groups, right? Right in the center there is your phosphate. So these phosphate little compounds bind to each other. And when these bonds break, boom, a lot of energy is released. And similarly, when these bonds are reformed, energy gets stored in that bond. So also become familiar with these 3 forms of adenosine triphosphate effectively. So when you have 3 phosphate compounds bound to the adenosine backbone, we call that adenosine triphosphate, 3 phosphates. If we're going to remove one of the phosphate groups to release energy, we're going to be left with just 2, right? We're going to call that adenosine diphosphate, which is abbreviated ADP. So diphosphate, di meaning 2. And then if we actually slough off that 2nd one, we're left with only 1 phosphate group. So we call that adenosine monophosphate, right? AMP, mono meaning 1. So please understand those 3 and understand how you get from one to the other. And then over here on the top right is just another way of writing this out, right? But as you build from AMP to ADP to ATP, we're storing energy here. So when you generate ATP, you're storing energy. And you can think of that, like you can think of anabolic processes as a way of storing energy, right? Like we said that to do any sort of anabolic work, we need to use energy. You could also think about it like energy is never created nor lost. You could think about it, maybe a more appropriate way to think about it is to say you're storing energy that way. Whereas when we break ATP down, and up here in the top right, when we break ATP down to ADP, we're releasing some energy. And when we break ADP down to AMP, we're releasing energy. So yeah, take some time here. It's pretty straightforward, hopefully, but take whatever time you need to have this make sense to you. All right, so then also we're continually in this conversation talking about metabolic pathways. So metabolic pathways are chemical reactions or clusters of chemical reactions that occur sequentially to achieve a particular goal. So again, when we talk about breaking down glycogen and then further breaking glucose down to release energy, that's actually like a 12-step process to break glucose down to release energy. And we'll look at that coming up. So oftentimes we sort of talk about an overall reaction, but when we say that, we're really talking about clusters of oftentimes like a half dozen or a dozen individual chemical reactions that make up this complete pathway. So yeah, an important concept to get to. And then mitochondria, we're going to see, especially as we talk about the metabolism of glucose, we're going to see that much of those processes occur in the mitochondria. So the mitochondria is one of your organelles inside of the cell, and you can think about it as like the little powerhouse, the little energy production organelle. So the mitochondria is the primary site of ATP production. And then we are also going to take a look towards the end of this lecture and see that macronutrients, they each have their own unique metabolic pathways to providing ATP or releasing ATP or building ATP, I guess ultimately is what we're doing here. So they each have their own metabolic pathways, but then they do have quite a bit of overlap. So what we're going to be talking about now is each of the 3 macronutrients. So we'll talk about carbohydrates, and we'll talk about lipids, and we'll talk about protein. And we're going to see how each of these three macronutrients has their own metabolic pathways, their own cluster of chemical reactions to produce ATP. And then we're going to see how they overlap each other. So this is effectively the content of this chapter. And so really critical for you to pay close attention on these coming slides, but really, again, spend whatever time you need to fully, grasp these concepts, because they can be a little heavy. So real quick, a nice picture to get us started. So this is a cell, right? Not quite some sort of weird 3D cross section here. So in the center, you've got the nucleus. And then we've got ribosomes. Remember, the ribosomes were important in building protein, that process of translation, in transcription and translation. And then you've got the endoplasmic reticulum, which can actually help in the final stages of protein manufacture. And then here you see the mitochondria. So the mitochondria being, this is the one we're going to talk about right now, the site of ATP production. And then we're also going to reference the cytoplasm, which is basically just the sort of liquidish interior of the cell. So we'll see that some of these metabolic processes take place in the cytoplasm, and then a lot of them take place in the mitochondria. Okie dokie. So I realize this is pretty small font. I guess I wanted to keep the girl up here showing, unfortunately she's eating like pizza and hamburgers and fries, but the point here is that when we eat, right, this is like a step of metabolism. So we break protein down to amino acids, we break carbohydrates down to monosaccharides, and then ultimately really glucose. And then we break lipids down, especially triglycerides, we break them down into glycerol and fatty acids. We're also going to mention alcohol in this chapter because alcohol does also provide calories. So we need to have a basic understanding then of how we metabolize alcohol, or even just know that we metabolize alcohol and it provides calories to the body. So again, this is an overview. So let's start with the carbohydrate. It's in the center, and it's really the one we're going to talk the most about in this chapter. So carbohydrates get broken down, right? And that process of breaking down is catabolism. They get broken down to glucose. Glucose further gets broken down into this molecule called pyruvate. This is all still catabolism. And then pyruvate gets further metabolized into acetyl-CoA. Acetyl-CoA enters this cycle of metabolic reactions. So still this complete metabolic pathway, major metabolic pathways, right? This concept here, metabolic pathways, is happening here on this slide. So acetyl-CoA goes through this chain of reactions, which we call the Krebs cycle, or the tricarboxylic acid cycle, the TCA cycle. And then ultimately, the little byproducts of all of these metabolic reactions enter into something called the electron transport chain. And it is here in the electron transport chain that we make a whole bunch of ATP. So I like this chart because the carbohydrate is right down the middle, because again, it's going to be the primary, carbohydrate metabolism is going to be the primary thing we talk about. But then let's take a look. I'll go purple for the amino acids. So protein gets broken down into the amino acids, and we're going to see we have we can classify amino acids in a lot of ways, but for this purpose, we can classify amino acids as either glucogenic or ketogenic. And let's pause right here, because I know these words sound monstrous, but just take a look. What does keto remind you of? Just anything. What's your first association? And what does gluco remind you of? Well, gluco might be easier. So hopefully glucogenic, hopefully that reminds you of glucose. So glucogenic amino acids are going to enter the carbohydrate metabolic pathways here at the end stage of glucose metabolism. So they're going to enter, glucogenic amino acids will enter at the site of pyruvate. So these amino acids can get metabolized into pyruvate. And then we'll actually see pyruvate can get reconverted back to glucose. So that's why we say that these are glucogenic, right? Glucogenic generate glucogenerating, these amino acids can generate glucose. So they're going to enter here at this site where glucose could be regenerated if needed. And then ketogenic, what does that remind you of? Keto, we talked about ketone bodies, we talked about ketosis and ketoacidosis. This has to do with fat metabolism, right? So ketogenic amino acids are going to enter metabolism down here at the site of acetyl-CoA, which if we go over to the lipid group, we'll see that fatty acids generate acetyl-CoA. So these amino acids relate more to fatty acids, and so they enter the full metabolic pathway here at the site of acetyl-CoA. So that's how amino acids affect metabolism. Like we said, there's overlap, right? The macronutrients overlap each other in the metabolic pathways to produce energy. So this is how they're overlapping. Again, I like to start with carbohydrate. I think it's kind of the centerpiece. So protein and amino acid metabolism overlaps with both carbohydrate metabolism and fatty acid metabolism. I probably should have looked at the fats first. So on the fat side, on the lipid side, right, we have glycerol and we have fatty acids. So these have a little bit more of their own independent metabolism relative to amino acids. So glycerol will also be converted to pyruvate. Remember, glycerol is a 3-carbon chain, and glucose is a 6-carbon ring, basically. So when glucose gets unfolded, you can make 2 3-carbon rings, which are very similar to the 3-carbon ring of glycerol. So glycerol can act a lot like glucose, so glycerol will be metabolized to pyruvate. And then fatty acids, right, these long chains of carbons, right, these will create acetyl-CoA. Fatty acids will be metabolized down into acetyl-CoA. And then alcohol will also enter at the site of acetyl-CoA. Okay, so this is just the overview, but good one to come back to, especially once we go through all of these processes. So I guess this isn't review, but let me go back again. When we talk about these clusters of chemical reactions, well, what are these chemical reactions? So some of these we've mentioned before, but not all of them. So I think I've got 4 slides here talking about these different chemical reactions. I'll write that up here. The chemical, and I'm going to abbreviate reactions, RXN. So the chemical reactions that make up these metabolic pathways. So we have dehydration synthesis and we have hydrolysis. Again, just look at these words and hopefully you'll have an almost immediate understanding of what they mean. Dehydration, right? To dehydrate means a loss of water, and to synthesize means to build something. So this is a building process, it's anabolic, and it's a process in which water is released as a byproduct, right? It gets dehydrated, so water gets released. So simple units combine to form a larger, more complex molecule. So it's anabolic, it's synthesis, and it's dehydration synthesis, so water gets lost or released as a byproduct. And then hydrolysis, again, hydro and lysis, right? Hydrolysis, hydro also meaning water, lysis meaning break apart. So hydrolysis, hydrolysis is a catabolic process. It's breaking down. And this is where a large complex compound is broken apart with the addition of water. So water is applied to break this apart. Hydrolysis, broken apart by the water. Again, pictorial representation here. So glucose and fructose joining together. When they join together, see they each have these little alcohol groups. 2 of those hydrogens and 1 of those oxygens are going to separate out to make water. So water is lost, and by losing that water, these 2 molecules, the glucose and fructose, can now bind to each other to form sucrose. So this is synthesis, right? Because we're building a larger, more complex. It's not that much more complex, but it is larger and it is more complex than individual glucose and individual fructose. So dehydration water is released in order to build a larger molecule, dehydration, synthesis. And then the same thing, we're going to look at this, but just reverse. So sucrose, now over here, we're going to start with sucrose. So we have this larger, more complex molecule. We're going to add water back in, and see the arrow implies that water comes in, whereas up here, the arrow implies that water is released. So follow the direction of the arrow too. So hydrolysis, right, we're going to apply water to break sucrose apart. So hydrolysis, sucrose gets broken down into glucose and fructose. Okay? You see the little plus sign in the middle, rather than these two compounds being bound to each other. And you'll see that each of these monosaccharides reformed their alcohol, their independent alcohol groups. Okay. Then we have this process called phosphorylation. And this one, I don't know if there's much way to break this apart, except that you'll probably notice phosphor, right? Phosphorus. So we have it again, a phosphate group, phosphorus group. And yeah, I guess I don't even know what elation means. There's probably an actual meaning there. So phosphorylation is the addition of a phosphate group to a compound. So actually, if I went all the way back to that ATP slide, where we had ATP, ADP, and AMP, triphosphate, diphosphate, and monophosphate, those processes of building the adding a phosphate group to the basic adenosine monophosphate structure, that's phosphorylation. I guess, why don't I go back just so we can see it? It's not that far. So this process, adenosine monophosphate, right? Adenosine monophosphate, if we just start with this one, but then we add the second one, that's phosphorylating, because you're adding this phosphate compound onto this adenosine monophosphate backbone. So AMP to ADP, that's phosphorylation. And then once you have these two, right, as is designated here by the ADP, once you have these two phosphate groups and you add this third one, that's phosphorylation. You're adding a third phosphate compound onto the structure that already exists. So from ADP to ATP, that's also phosphorylation. Right, and remember, it's actually in this phosphorylation, when we phosphorylate something, we're storing energy that way, right? So in the same way, when we dephosphorylate something, so I should write that here because that's the reverse, dephosphorylation, right? Dephosphorylation is pulling the phosphate group back off. So when, so when the high energy phosphate bonds in ATP are broken, we release energy, right? So let's go back. That's what's shown up here. When you start with ATP, when you start with the full adenosine triphosphate molecule, when you break off a phosphate group, we wind up with adenosine diphosphate, and this little independent phosphate group is, well, detaching this phosphate group from ATP is releasing energy, right? And the same thing when we detach the second phosphate group from adenosine diphosphate to render AMP, this is releasing energy, right? So energy is released when we break the phosphate bonds. And then again, energy and matter are neither created nor destroyed, so the phosphate group is always going somewhere else. So when one thing gets phosphorylated, something else gets dephosphorylated. So we're going to see actually, well, we won't really in this class, but if you go on to study nutrition, which would be cool, you'll see that glucose does actually get phosphorylated in this long process of glucose being converted to pyruvate and ultimately acetyl-CoA. There's actually a lot of phosphorylation and dephosphorylation happening in this process. Okay. And then the golden one here are oxidation reduction reactions. So yeah, these are all important, but I think redox is maybe the most complicated to understand, but it's not too bad. It's pretty straightforward, actually. So in oxidation reduction reactions, we're looking at a transfer of atoms and specifically their electrons. So in any case, we have two reactions that occur at the same time. So anywhere you have oxidation, you also have reduction, which is actually the same here. Anywhere you have phosphorylation, you necessarily have dephosphorylation. And really, even we could almost even say the same thing here. Anywhere you have dehydration synthesis, you're likely to also have hydrolysis and vice versa. But especially here in redox reactions, so we even call these redox, is the abbreviation for oxidation reduction, because they go together so intimately, right? So we instead of calling it oxidation reduction, we just kind of call it one term, redox, but that's also shorthand for oxidation reduction. Okay, so let's talk about the two processes individually so you understand them. So oxidation is when one molecule is donating an electron, and particularly usually donating an atom. Usually this atom is hydrogen. So its electron is removed by oxygen. Yeah, we're going to see that oxygen is commonly the atom that does this process of oxidation. So something else gets oxidized by oxygen. Hence this word being oxidized, oxidation, because oxygen is the atom doing this electron taking. So oxidation, there's this little mnemonic called oil rig. And maybe if you learned about this in basic bio, this will ring a bell. So oil, this is abbreviated as oxidation is loss, right? So if something is oxidized, it loses an electron, whereas reduction is gain. So the rig means reduction is gain. So if something is reduced, it actually gets an electron. So oxidizing is losing an electron, reduction is gaining an electron. So let's look at a schematic here. So oxidation reduction, this is kind of a complex molecule to take a look at, but we are going to see this FADH2 molecule quite a bit. And we're going to revisit this when we start to talk about B vitamins, which I believe is chapter 8. So here we go. So we have this molecule FADH2, right? And just by the name of it, you can see that it probably has these two random hydrogens. Well, not so random, but these two hydrogens attached. And here they are in the chemical drawing. So when this molecule gets oxidized, remember, what is oxidation? What is it? Oil. Oxidation is, what's the L for? Loss, right? Loss of what? What are we losing? Our brains, our minds? No, we're losing electrons, right? So oxidation is loss of electron. And so here it is. Remember, follow the arrow. The arrow is going away. So that means these things are coming out of the reaction. 2 electrons. And if you don't remember from basic chemistry, that E with the negative sign, that's the abbreviation for electron. And generally, wherever these electrons are, 2 little hydrogen atoms, wherever there's an electron being lost, usually there's technically a hydrogen atom being lost. And so that's right, we saw FADH2 has these 2 hydrogens that get lost as this molecule becomes FAD. This nitrogen no longer has that hydrogen attached, right? And this nitrogen also no longer has that hydrogen attached. So FADH2 gets oxidized to become FAD, and it loses electrons. And then in the same way, when we go purple, I guess, we'll see that reduction is the reverse of that process. So FAD can get reduced to FADH2. And remember, what is reduction? Rig, right? Reduction is gain. Gaining what? Gaining knowledge, gaining electrons. So FAD gets reduced to FADH2. And here again, follow the arrow. You see these two electrons, which are always accompanied by these two hydrogens. These are coming into the reaction. So that means they're going to bind back to this FAD compound, making it FADH2. So oxidation reduction, it's basically just this whole process of transferring electrons, which usually go along with hydrogen atoms, right? Now it's funny here because we said that, so we said that the electron is often removed by oxygen. So what we're not seeing here is that these electrons are going to go to some oxygen containing compound, right? Or, potentially making water even, right? H2O would be water. So sometimes we see all of these reactions, dehydration, synthesis, hydrolysis, phosphorylation, redox reactions, these are often overlapping reactions as well. But particularly, we're going to look at oxidation reduction reactions. Let me go back here. We're particularly going to see a lot of redox here at the site of the electron transport chain, and also here at the site of the carboxylic acid cycle. So that's where we're going to see a lot of redox reactions. But really all throughout these major metabolic pathways, we're seeing, well, let's write it out. We're seeing dehydration. I'm going to abbreviate dehydration synthesis. We're seeing hydrolysis. We're seeing phosphorylation and dephosphorylation, and we're seeing redox. So throughout this entire blue area, we're seeing these major metabolic pathways, which are made up of these individual chemical reactions, right? That's what we said here. These major metabolic pathways are made up of these clusters of chemical reactions that occur sequentially to achieve the particular goal of either converting glucose to pyruvate or converting acetyl-CoA, sending that through the TCA cycle, and generating all of these electron transport compounds. So that's where all of these fit in, is in these larger metabolic pathways of breaking our macronutrients down to yield energy. So let's also talk about a few enzymes, a few, I guess, classifications of enzymes that we're going to see. So we can talk about, and just a review again, we've never written this out yet in this class, but enzymes are generally proteins that help chemical reactions go. Enzymes, we often say, speed up chemical reactions, or here it's written that they mediate chemical reactions, like they're kind of a referee. Probably isn't a good analogy, but I guess you could think of like a referee, right? Enzymes, but they're even more necessary than a referee. Like without the enzymes, the chemical reactions won't happen. Okay, so two subclasses of enzymes that we'll talk about here for our metabolic processes. We have coenzymes and cofactors. So coenzymes, let's see. Yeah, so coenzymes, they're non-protein substances, right? So I just said that enzymes are proteins that support the enzyme activity. Typically, we'll see a lot of vitamins that function as coenzymes. We're also going to talk about FAD and FADH2, but we're going to see that they are built from vitamins. Whereas a cofactor, like this, I guess let's look at it this way. So a enzyme is a molecule. It's a more complex molecule. Whereas a factor, you could think of it as something a little simpler, like a mineral, right? So vitamins, you might remember from Unit 1, vitamins are more complex. Minerals are already at their basic elemental form. Remember when we talked about vitamins being able, like vitamins can be broken down by heat or acid or pressure or light, whereas minerals can't. Like any cooking processes that we might do, they're not going to alter the shape or the form of a mineral because the mineral is already as simple as it can be. So a coenzyme is typically a vitamin, a cofactor is typically a mineral. Other than that, both of them support the function of the enzyme. So some common cofactors that we'll see: iron, magnesium, and zinc. And again, these are all minerals. So coenzymes and cofactors support the enzyme activity, right? Necessary for enzyme activity, required for enzyme activity. It's just that there are two different ways that we can support the enzyme activity. So here is an example. Do we have a coenzyme here? I guess it's not going to show a coenzyme, but we're just going to show the cofactor. But they look a lot the same anyway in practice. So up top, you see, here's your inactive enzyme, right? It's this large protein compound. It has a specific shape to fit whatever chemical compound it's going to support in chemical reaction. You'll see this kind of looks like a tooth. This chemical compound won't fit into this shape. So the compound cannot bind to the enzyme, so the chemical reaction will not take place. Whereas in the bottom half, we now have entered the cofactor. So it's the same enzyme, right? I don't know why they changed the color, because I guess now it becomes active, right? So we call this an inactive enzyme. And now once the cofactor is bound, we call this an active enzyme. And so now you'll see the only space left for binding is this little area. And so again, same chemical compound. Now this chemical compound, this little tooth-like thing, will actually fit here. You'll see the bottom shape matches that like a puzzle. And so now you actually have a complete, I think this is what we call a hollow enzyme, right? You have a complete enzyme compound structure, so the chemical reaction can take place. So you can also think about coenzymes and cofactors as being, as being necessary to convert the inactive enzyme to the active form. So without the cofactor, without the coenzyme, you just have an inactive enzyme. So it's effectively useless until you have the cofactor or the coenzyme. Okie dokie. If you need a break, this is probably a good place to pause because now we get into the thick of it. So now we're going to talk about how do we extract energy from carbohydrates, and then we'll talk about lipids, and we'll talk about proteins, and then we might be done with chapter 7. So let's see. A little review here, maybe not. When glucose is transported to the liver, right, this happens generally after a meal. We're going to see that glucose gets phosphorylated and then metabolized. So remember, metabolism is both breaking down and building up. Metabolism is both catabolism and anabolism. So if you just say metabolism, it could be either breakdown or buildup. So in this case, glucose will get either metabolized for energy, broken down, that's catabolism, or will get stored as glycogen, that's anabolism, that's building glycogen. Or glucose could get released back into the bloodstream to travel to other cells to be used as fuel, again, metabolized for energy, broken down, or stored as glycogen like in your muscle. Again, that would be anabolism. And then if glucose exceeds the current energy needs of the body, then glucose will be converted to fatty acid and stored as triglyceride in adipose tissue. So that's also anabolic, storing fat, building your fat tissue. And then remember we said that fructose and galactose, your other monosaccharides, these just get converted to glucose at the liver and follow these same pathways depending on what the body needs. Okay, glycolysis. Oh, that's funny. Okay, glycolysis is the breakdown of glycogen, right? Glyco meaning glycogen and lysis meaning breakdown. So it's again a series of chemical reactions that converts, sorry, that's glycogenolysis. Sorry, back up. So glycolysis, right? Glycolysis is referring really to glucose breakdown. Okay, that was a big error. Okay, so get that straight. Sorry, glycogenolysis breaks down glycogen and releases glucose, but then the process of breaking glucose down is glycolysis, glycolysis. Sorry, that was maybe the biggest misspeaking that I could make. Okay, so glycolysis is literally converting glucose to pyruvate. I'm just going to write that there just to really address my error. Glycolysis equals glucose being broken down to pyruvate. Right? In particular, glycolysis, when we break glucose down to pyruvate, we actually take one glucose molecule and render two pyruvate molecules. That's an important piece to know as well. Glycolysis takes place in the cytosol. Remember that image of the cell we saw? We're not in the mitochondria yet. This process just takes place in that liquidy middle substance area of the cell. This is also anaerobic. This is kind of a new term to us. Anaerobic meaning no oxygen, right? Aerobic meaning that we need oxygen. Anaerobic meaning without. So anaerobic chemical reactions meaning without oxygen. We're going to see that a net of 2 ATP will be used as energy for the cell. Meaning a net of 2 ATP actually go into this process of breaking this down. No, I said that backwards. That's weird. A net of 2 ATP will be rendered for use by the cell. So we're going to basically create a net of 2 ATP. For my class, I know sometimes in biology classes or physiology classes, the instructor wants you to know the different numbers of ATP molecules being produced at different areas. I'm not going to ask you the precise number at different areas, but I do want you to understand that carbohydrate metabolism generally yields fewer ATP than lipid metabolism. So that's the major takeaway I want you to get from ATP production. So if it helps remember the rough number of ATPs, then I certainly won't stop you from doing that. But you don't need to know, right, that 2 ATP are used here and 4 ATP are created here, thus yielding a net of 2 ATP produced for the cell. Okay, so glycolysis, remember I said it's actually a 12-step process, but we are really only we're gonna condense it down into one step, right? I want you to basically know that glucose, right, which is, we started with a hexagon of six carbons, right? But here they are now 1, 2, 3, 4, 5, 6, just laid out in a chain so that they can be more easily broken apart. So we're going to have 12 processes, boom, 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 of converting glucose down into these 2 pyruvate molecules. So back here, when I said that glycolysis is taking 1 glucose and making 2 pyruvate, that's what I mean. Glucose starts with 6 carbons, right? 1, 2, 3, 4, 5, 6. Pyruvate, 1 pyruvate molecule, this is 1 pyruvate molecule. It's a 3-carbon molecule, so if you start with 6 carbons, you can make 2 3-carbon chains. So we make 2 pyruvate. I ask that question on exams, and I always try to trick you, and I give you the option to select 1 pyruvate or 2 pyruvate. So make note that it is 2 pyruvate. And then just to point out these different chemical reactions, right? We talked about phosphorylation and dephosphorylation. So you'll see ATP gets used right here. Adenosine triphosphate gets used in this conversion of glucose to glucose 6-phosphate. So remember, ATP is going to slough off one phosphate group, so ATP is going to get dephosphorylated, yielding adenosine diphosphate, but that phosphate group goes somewhere. Remember, we said wherever we see dephosphorylation, we see phosphorylation. So in this case, the glucose molecule gets phosphorylated to become glucose 6-phosphate. So technically, actually, the 6th carbon in the glucose molecule will suddenly get the addition of a phosphate group, so glucose 6-phosphate. And then on down, you can see that we get another phosphorylation, dephosphorylation happening. We get a lot of phosphorylation, dephosphorylation happening in this process of glycolysis. And again, sorry, just for kicks, I'm going to go all the way back here. Remember, we said major metabolic pathways. We said we're going to see phosphorylation and dephosphorylation, and we're right here in this process of glucose getting converted to pyruvate. We're seeing a whole lot of phosphorylation and dephosphorylation here. Okie dokie. Okay, so glycolysis. So the end product of glycolysis is what? You should definitely be able to know that. If that's it will probably be a test question or a quiz question. What is the end product of glycolysis? The end product of the glycolysis is 2 pyruvate per 1 glucose. Okay, so then the pyruvate has 2 fates, right? Pyruvate can do 2 different things, depending on whether we have anaerobic conditions or aerobic conditions. So under anaerobic conditions, because remember glycolysis is happening anaerobically, there's no oxygen here. So under anaerobic conditions, if there continues to not be enough oxygen, then pyruvate gets converted to this molecule called lactate. Oh, and what do we have here? Looks like it might be a redox reaction, right? This is a redox reaction happening to convert pyruvate to lactate. So under anaerobic conditions, pyruvate will make this molecule lactate, and lactate is basically like energy in waiting, if you will. And yeah, so it might be helpful to think about when do we have anaerobic conditions. This would be like really high intensity exercise where your respiratory system, your breathing, can't keep up with the amount of energy that your body is using. And so in this process of trying to make energy for your working muscles, there's going to be a little bit of a backlog. And so this backlog is lactate or lactic acid buildup. And it's going to be until your body stops working at such a high intensity that you have this backup of lactate or lactic acid. So once you're done with the exercise, your body will start converting lactate back to pyruvate, back to glucose, so that the glucose can then be used to be broken down and supply energy. So lactate is kind of like a holding place for energy until you get enough oxygen coming in to manage your body's production of pyruvate. So yeah, so let's start down here. So in the working muscle, if you don't have enough oxygen, so not enough oxygen, pyruvate, glucose is going to make pyruvate, but then pyruvate is going to get converted to lactate. Lactate is transported to the liver. So let's say this is you're exercising over here, right? And now you're going to rest afterwards, right? So now lactate is going to get transported back to the liver. Now you do have enough oxygen. So the liver is going to convert lactate back to pyruvate and then either break the pyruvate down further, as we'll see on the next slide, and produce more ATP, or potentially convert it back to glucose and store the glucose as glycogen. So you always have this, you don't always, but especially after high intensity exercises, you'll have this little lactic acid cycle where you'll build up some lactate during the exercise if you don't have enough oxygen, if it's a really high intensity. And then as soon as you're done and you're resting, your body will be able to reuse that lactate, right, and actually break it down. So typically our bodies will use up lactate within usually within 45 to 60 minutes after an exercise. So a lot of people think that we still carry around all this lactic acid the next day, but actually we use it up pretty efficiently once we're done exercising. Okay, so the other fate of pyruvate under aerobic conditions, right? So like all of the time when you're just normally at rest, or maybe you're exercising, but it's not such a high intensity. Pyruvate is going to get converted to this compound called acetyl-CoA. But remember, you have two pyruvate, which are 3 carbon molecules. Acetyl-CoA down here at the bottom is a 2 carbon molecule with this CoA group attached to it. So 2 carbon with CoA. So if you have 2 pyruvate, very easily you can make 2 acetyl-CoA. You'll see that the pyruvate molecules lose 2 carbon dioxides. That's where we get this idea of oxygen coming in and CO2 coming out, right? That's basic respiration, right? O2 in, CO2 out. So here's where some CO2 is produced, is when pyruvate gets converted to acetyl-CoA. Because pyruvate has 3 carbons, acetyl-CoA has 2 carbons. So obviously each pyruvate needs to lose one of its carbons, and it does so by forming carbon dioxide. So this process is happening here, right? When the lactate gets converted back to pyruvate, this pyruvate can then get converted to acetyl-CoA. This process is also happening, as I said, just under normal circumstances, normal conditions, when there is enough oxygen present. So this word aerobic means oxygen is present. Right. Okay, so I'm also going to back way up to here. I missed it. Right, so we're here, right? We took glucose, we converted it to pyruvate, and now we're going to convert pyruvate to acetyl-CoA. So we're still right in here. Okay, so we made 2 acetyl-CoA, and now, we'll come back to this one. And now acetyl-CoA is going to enter the tricarboxylic acid cycle, or the Krebs citric acid cycle. You can call it by either of those names. The tricarboxylic acid cycle, I guess I should write that out. Tricarboxylic acid cycle, or sometimes we call it the Krebs. Oops. Sometimes we call it the Krebs citric acid cycle. So you can call it the TCA cycle, you can call it the citric acid cycle, you can call it the Krebs cycle, you can call it the Krebs citric acid cycle. Any of those names will do. And so you'll see part of why it has this citric acid name, or tricarboxylic acid name, is because the first metabolite is citric acid. So that's where these names come from. Krebs also is probably the name of the person who invented it, probably a man, I guess. Okay, so 2 acetyl-CoA enter the tricarboxylic acid cycle. You do not need to know any of these intermediate metabolites, but again, this is what we mean when we say metabolic pathways are these series of individual chemical reactions. Each time you see an arrow, that's indicating a chemical reaction, or sometimes, as would be up here in glycolysis, one arrow is actually representing 12 independent chemical reactions. So what I do want to highlight here is hydrolysis, right? The addition of water. We have hydrolysis happening. We have redox reactions happening. right? The transfer of electrolytes. Electrons. I should have taken a break. The transfer of electrons, and you'll always see that with the transfer of hydrogen atoms. So we don't so easily see the transfer of the electrons, but we can always identify the transfer of the hydrogen atoms. So again, here, remember this is the one we looked at, is the conversion of FAD to FADH2. So see if you can remember, right? If FAD becomes FADH2, what is happening to FAD? Is FAD gaining or losing electrons when it becomes FADH2? It's gaining. And what did we say is gaining? We said reduction is gain, oil rig. So FAD here gets reduced to FADH2. Also here you see a phosphorylation reaction happening, right? GDP gets phosphorylated with the addition of this phosphate compound. Okay, so yeah, I guess the big takeaway I want you to have with the tricarboxylic acid cycle is redox, actually. I do want you to, so let me change to like red. I do want you to know, and actually let me just erase all of this. So I do want you to know there's a whole lot of redox happening here. And the point of that is because all of these compounds, these NADH molecules, these FADH2 molecules, there's a third type, but it's not shown here. So these redox reactions, we're generating a lot of NADH, we're generating a lot of FADH2. And why this is important is because they're holding on to these additional hydrogen and therefore these additional electrons, right? So we're basically building up this supply of these molecules that are carrying around a bunch of extra hydrogen and a bunch of extra electrons. So that becomes important when we then move to the electron transport chain. Because now what we're doing is we're taking all of these NADH molecules, we're taking all of these FADH molecules, and you see this line up here, these lines are showing that these NADH and these FADH2 molecules are coming from the TCA cycle, right? The TCA cycle, right? So all of these NADH molecules, all of these FADH2 molecules are generated here, and then they play a significant role in the electron transport chain because they all have all these extra electrons to donate. So they're donating electrons in this chain of events, in this chain of reactions that then supports the creation of ATP, 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 ATP. So all these electrons get stored up here in NADH and FADH2 in the TCA cycle, and then they donate their electrons in the electron transport chain so that we can build ATP. So the primary purpose of the electron transport chain is to generate ATP. And I don't know if I have it written. No. So usually the full process of carbohydrate metabolism yields somewhere like 36 to 38 ATP. Again, I don't need you to remember that number, but I want you to remember that that's going to be a smaller number than the number of ATP that gets generated from fat metabolism. So this here ends our discussion of carbohydrate metabolism, at least temporarily. And this is also a decent page to just see the overview, right? We start with glucose. The process of glycolysis takes us to 2-pyruvate. And then we can make 2-acetyl-CoA from the pyruvate. Acetyl-CoA initiates the tricarboxylic acid cycle. The tricarboxylic acid cycle creates all these reduced compounds that carry a bunch of extra electrons, and then those, and these are actually coenzymes. I'm sorry. If we go way back here, this coenzyme page, right? Coenzymes, FAD and FADH2, and NAD and NADH. These are coenzymes, so they're helping to support all of these reactions that happen in the electron transport chain. And support in part by donating their electrons. And so in the electron transport chain, all those electrons get donated so that we can create all these ATPs. So that's the overview of carbohydrate metabolism. Let me just look at this. So again, I feel like this is way out of order again for some reason. But this is just, again, another review of the metabolic crossroads. So where the substrates from all of the different macronutrients come into play, right? Glucose, amino acids, fatty acids, glycerol, and alcohol. They're all entering either at pyruvate or at acetyl-CoA. Do you see that, right? All roads lead either to pyruvate or they lead to acetyl-CoA. Those are the two entry sites for our building blocks of our macromolecules, of our macronutrients, to come into this process of metabolism. Okay, so now let's talk about fat metabolism. So another word here to know is lipolysis. So you can think about glycolysis and lipolysis as similar terms, right? Lipolysis is the breakdown of fat. So lipolysis is where we take dietary or stored, adipocyte, this is a fat cell, adipo meaning adipose and cyte meaning cell. So adipocyte, a fat cell, triglycerides. So we take triglycerides and break them down using lipases, right? Enzymes that break down lipids to yield glycerol and 3 fatty acids, right? That's the process of lipolysis. Same way that glycolysis is the breakdown of glucose. And then as we've seen, glycerol can get converted to pyruvate, and then pyruvate can get converted to acetyl-CoA, and then enter the TCA cycle. And then these fatty acid chains, they ultimately will make acetyl-CoA. So this process of breaking down the fatty acids this is an important term to know, is beta oxidation. Lipolysis is also an important term to know. So beta oxidation is fatty acid oxidation. It's the metabolic reactions that oxidize fatty acids. And in that process, we produce water, carbon dioxide, and ATP. So beta oxidation is the metabolism of fatty acids to produce energy. Dang, I'm sorry. These are, well, okay, no, sorry, this is not out of order. So here's the glycerol getting converted to pyruvate. This is actually also a helpful overview. So you start with the triglyceride, right? The glycerol backbone and the 3 fatty acids. We need lipases, enzymes to break down lipids. And of course, it's going to break down into glycerol and then free fatty acids. The glycerol, this happens in the liver. The glycerol gets converted to pyruvate, and then pyruvate can get converted to acetyl-CoA, or as I said, pyruvate can actually get converted back to glucose. So remember, part of why this happens in the liver is because generally we're using glucose as our energy substrate, and we don't have a way, so all cells are set up to metabolize glucose, but not all cells are set up to metabolize triglyceride, right? So the liver, and actually the muscle cells, muscle tissue, but especially the liver, can take triglyceride, so the body has to get triglyceride out of storage, out of the adipose tissue, transfer it to the liver. Well, at the adipocyte, we can break the triglyceride down into glycerol and fatty acids. And then the glycerol will travel to the liver, and the liver can do the job of converting glycerol to pyruvate. And then depending on what the body needs at that time, if we just needed glucose, like to raise blood glucose levels, then we can convert pyruvate to glucose. But if we are using this for energy, then the pyruvate will get converted to acetyl-CoA, and acetyl-CoA will enter the TCA cycle. And then the fatty acids we'll talk about separately. So the fatty acids will also get released into the bloodstream, and they'll go again either to the liver or to working muscle tissue. So yeah, so here when we say that fatty acids get released into the bloodstream, remember fatty acids are fat. They're fat soluble. They're not water soluble. So we need albumin. This is a protein in the blood that transports fatty acids. It's the fatty acid taxi. Albumin is the fat taxi. So fatty acids get transported via albumin in the blood, and they get delivered to different working cells throughout the body that need energy. So again, this is going to be either muscle or liver, right? We not all of our body tissue, not all of our organs can break down fatty acids. Really, primarily only our muscle tissue and our liver can break down fatty acids. So that's going to be their destination, either muscle or liver. And then once inside the cell, the muscle or the liver cell, fatty acids have to be activated by something called coenzyme A before they get brought into the mitochondria. And there's also another transport protein called carnitine that transports the fatty acids into the mitochondria so that they can then undergo this process of beta oxidation. So yeah, so ultimately these fatty acid chains get broken down into 2 carbon segments that then form acetyl-CoA. So great, here's an excellent schematic of this. This is also a super critical thing to remember. So, or to understand, I guess. So, and I think it's actually very understandable. I don't think there's a lot that you need to remember here. So here's a fatty acid chain, right? And we've got 16 carbons, and this particular fatty acid is called palmitate. That's a type of fatty acid. So it's 16 carbons, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, right? So the process of beta oxidation is literally this, sloughing off these 2 carbon segments or cleaving off 2 carbon segments. So as we cleave off this 2 carbon segment, okay, it's shown here. So this, let me change the color, I guess. This 2 carbon segment gets cleaved off and joins with this CoA. So see CoA coming in here. This come over here now. So now you have this 2 carbon. So these 2 carbons come here, and this CoA joins with these 2 carbons, and you make acetyl-CoA. The same acetyl-CoA that we were talking about right here. The same acetyl-CoA. This acetyl-CoA, right? Fatty acids make acetyl-CoA. Glycerol, as we just saw, basically makes pyruvate. And then pyruvate will either make acetyl-CoA or make glucose, right? We just saw that here. Glycerol, only fate is pyruvate. But then pyruvate has 2 fates, either glucose or acetyl-CoA. Fatty acids, only 1 fate, which is acetyl-CoA. I don't know why they don't write that. Acetyl-CoA. Great, so let me go back to yellow. So now if we just cleaved off 2 carbons, now we have a 14 carbon chain, right? So now we only have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Now we only have 14 carbons. So another CoA molecule comes in, right? Another CoA molecule enters. These last 2 carbons get sloughed off, and now we are left with, here goes another acetyl-CoA molecule. We create another acetyl-CoA molecule, and now we're left with a 12 carbon chain, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Enter another CoA molecule, slough off these last 2 carbons. You'll make another acetyl-CoA, right? And now we're going to be left with a 10 carbon chain, right? And so you can do this all the way down until you no longer have a fatty acid chain, right? Because from 10, we'll make another acetyl-CoA and we'll be left with 8 carbons. And then from 8, we'll make another acetyl-CoA and we'll be left with 6 carbons. And then we'll make another acetyl-CoA, we'll be left with 4 carbons. We'll make another acetyl-CoA, we're left with 2 carbons, and we make a last acetyl-CoA. So what's really important to note here is, right, we started with 16 carbons in the chain. And so we're making acetyl-CoAs, which are 2 carbon compounds, right? So what's 16, how many 2 carbon compounds can we make if we start with 16, right? Well, basic math, we can make 8 down here at the bottom. Ah, shoot. Dang, sorry about that. Let's just make sure I didn't get booted here. Okay. So, sorry, I must have hit a button, which is fine because that was a big mess. So, right, if you start with 16 carbons and you're making these 2 carbon acetyl-CoA molecules, right, how many 2 carbon acetyl-CoA molecules can you make? You can make 8 of them. So the same thing, if you had a 22 carbon chain, you could make 11 acetyl-CoAs, right? And if you had a 14 carbon chain, you could make 7 acetyl-CoAs. So really, in particular, this is what I want you to understand. If we have 8 acetyl-CoAs, right, ultimately, all these acetyl-CoAs are going to run into this carboxylic acid cycle, right? Remember I said before that we're going to generate roughly 36 ATP. I said 36 to 38, but let's go with 36. I said that from 2 acetyl-CoA, we're going to make about 36 ATP. So that means 1 acetyl-CoA would make roughly, what, 18, right? 18 ATP. So now, look at this, from 1 fat molecule, if we have 8 acetyl-CoA going through, and we're saying that roughly 1 acetyl-CoA makes 16 ATP per acetyl-CoA. And if we have 8 of them, what's 8 times 16? 80 plus, what, 48? So 128. So 8 acetyl-CoA, so 1 fatty acid is going to make roughly 128 ATP. So I don't want you to know these numbers. These are really rough numbers because there's actually there's always slight variation. I really don't care about the numbers. I just want you to know that 128 is a much larger number than 36. So what am I saying there? I'm saying that 1 glucose molecule makes about 36 ATP. That's some energy, right? But 1 fat molecule makes a whole bunch. And actually, this isn't even a full fat molecule. This is only 1 fatty acid chain. Now we have 3 chains, right? We have 3 chains per triglyceride. We could even multiply 128 by 3, and let's just call it 400, right? 375. So 1 fat molecule, 1 triglyceride, is going to make significantly more ATP than 1 glucose molecule, right? So the reason that I'm emphasizing this is because when we say that fats are more energy dense, right, we say that fats provide 9 calories per gram, whereas carbohydrate and protein only provide 4 calories per gram. This is why, because fats contain so many more carbon-carbon bonds relative to a glucose molecule or an amino acid backbone. So, yeah, make note of that whole. I know it's a big process, but if you can, make note of that. Right, we're making a whole bunch more acetyl-CoA than 1 glucose molecule can make. But you'll notice we're also going to make the other final products here are also going to be these coenzymes, right? So we still have this same fate of acetyl-CoA. Fatty acids can also make acetyl-CoA, and acetyl-CoA is still going to enter the TCA cycle, right? And then ultimately the electron transport chain. So that's what happens after this step. Oh, good, there's a chart. So here's the full triglyceride metabolism overview. So you start with the triglyceride, you break it down into its component parts, glycerol and fatty acids. Fatty acids, as we just saw, undergo this process of beta oxidation. They yield a whole bunch of acetyl-CoA, and then all of those acetyl-CoA enter the TCA cycle, forming these coenzymes. And then the coenzymes enter the electron transport chain and generate a whole bunch of ATP. And then also that glycerol, so glycerol is also going to contribute one acetyl-CoA, right? And it's going to have the same fate. So ultimately building all these ATP molecules, extracting all this energy from the triglyceride. Okay, so then this is also a little image to help you understand when I said that from a triglyceride or from a fatty acid, we yield so much more acetyl-CoA, and therefore we can make so much more ATP than from one glucose. So here's a pictorial representation of that, right? So the glucose only has 6 carbons. The palmitate has 16 carbons, right? So remember, one glucose, that can make 2 acetyl-CoAs. How many acetyl-CoAs can we make from a 16 carbon? 8 acetyl-CoAs, right? So we have 8 versus 2. 8 is a much larger number than 2, so we're going to yield a lot more ATP, so a lot more ATP relative to the glucose. Okay, so then we also need to talk a little bit about this production of the ketone bodies. So we have all this whole lovely process. Oh, I missed that slide. So we have this whole lovely process, right, of building a bunch of acetyl-CoA from glycerol and fatty acids. But we're also going to be making these ketone bodies. So I'm going to go back to that other slide in a second. I didn't realize I skipped it. So ketones, or ketone bodies, as they're sometimes called, are byproducts of fat catabolism, fat breakdown. So it's not really shown on any of the diagrams, unfortunately, but in these processes, additional ketone bodies are produced, basically, right? And so these ketone bodies can provide energy, but it's metabolically inefficient, basically. So ketone production is basically a backup source of energy for when cells become deprived of carbohydrate. So again, it's not the body's preferred way of providing energy, but it's just a backup system, because sometimes, especially throughout human evolution, access to food was not always, and it's still for a lot of people around the world, access to food is not always reliable. So there has to be some sort of backup energy production system, and that is this process of ketone body production. So the issue with ketone synthesis is only when it goes on too long unchecked. We can wind up with this state of ketosis, which is where these ketone bodies, which are very acidic, start to inappropriately lower our blood pH, right? So a lot of people are into the whole keto diet, the ketogenic diet right now. And that under medical supervision, that can be a way of managing particularly epilepsy. But we do not have much information at this point in time to indicate that long-term ketosis is safe for anybody. So we have a lot of people following a keto diet right now, most people doing it without medical supervision. From my viewpoint, I see a lot of people thinking they're doing ketosis, but they're actually just doing a lot of protein, so they're probably not actually getting into a state of ketosis. But some people, I'm sure, do manage to get into ketosis. But the issue is, if this goes on for a long period of time, like more than 2 weeks even, you can run into this issue of ketoacidosis, where the pH of the blood falls too far and can result in severe dehydration and potentially even a fatality, even death, because we can't exist with a very acidic blood pH. So ketoacidosis is where the blood becomes too acidic to be healthy. Again, most people out in the general public who think they're following a ketogenic diet probably are not actually getting into a state of ketosis. It's very hard to do, right? It's not something the body is trying to do. It's a backup under severe starvation, actually. So in order to induce ketosis, that's why we generally need medical supervision to actually look at these metabolic processes happening in the body to see that we're actually getting the body to a state where ketosis is occurring. So we can also see ketosis and ketoacidosis in diabetes, where that we have carbohydrate deprived cells, right? Type 2 diabetes and even type 1 diabetes, we can have carbohydrate deprived cells because we have some issue with insulin, right? In type 1, there might not be enough insulin, and insulin's job is to get the cells to take up glucose. So if we don't have enough insulin, cells can't take up glucose, so they will become carbohydrate deprived. In type 2 diabetes, we have insulin usually, but the cells are insulin resistant. They're not responding to insulin. So again, if insulin's job is to have the cells take in glucose and the cells are ignoring insulin, then we're going to have carbohydrate deprived cells. So ketoacidosis can actually also become a major problem for unmanaged diabetes. Alrighty. And now our third macronutrient. Oh, sorry, let me go back here because I skipped this slide. So what this slide is saying is that no metabolic pathway can convert acetyl-CoA to pyruvate. And this is important. So going back here, I guess let's go actually all the way back here. So remember we said that pyruvate can make acetyl-CoA, or it can backtrack and make glucose. There's no way that a fatty acid could ever make glucose, right? The fatty acid can only ever make acetyl-CoA. Let me see if we even have, let me add right here, I guess, so we don't have to backtrack too far. Right here, pyruvate can backtrack. This process is reversible. Glucose can make pyruvate, pyruvate can make glucose, right? We saw that with with the lactic acid cycle, right? Pyruvate, glucose can make pyruvate, and then again, pyruvate can make glucose, right? So this process of glucose to pyruvate and pyruvate to glucose, it's reversible. Let's say it again. Oh, here, it's drawn actually here. So what we're saying, what this other slide is saying, is that fatty acids can never become glucose. They only make acetyl-CoA. Glycerol, which is the other component of a triglyceride, right? Glycerol and 3 fatty acids. Glycerol can effectively become glucose because glycerol can make pyruvate, and pyruvate can then be converted back to glucose. But from a fatty acid, we can never get back to glucose because acetyl-CoA cannot backtrack and become pyruvate. This process of pyruvate to acetyl-CoA is not reversible. So what we're saying here is that cells cannot convert acetyl-CoA back to glucose. It's impossible for fatty acids to feed into glucose production. And I don't know why it's not written here. A reason for why this is important is because right here, there's actually, it's not drawn here either, but there's actually another metabolite that acetyl-CoA binds with to start the citric acid cycle. And that metabolite derives ultimately from glucose, or you could say derives from pyruvate. So there's this concept, we say fats burn in a carbohydrate flame. I guess I'll write fats burn in a carbohydrate flame. Meaning that in order to fully metabolize all of these fatty acids, we actually need glucose. We need carbohydrate in order to fully metabolize all of these fatty acids, right? In order to fully metabolize all of the acetyl-CoA that these fatty acids make. Alrighty, so now let's talk about energy from proteins. So we don't have a fancy term like glycolysis or lipolysis, and that's in part because proteins are so complex, also in part because our bodies preferentially use fat and carbohydrate as fuel, and protein for fuel is really like a last resort. So protein is saved. It's really those amino acids are precious. They're saved for metabolic functions that literally can't be carried out by anything but protein. So again, enzymes, hormones, immune cells, cell communication compounds, cell membrane transporters, blood transporters, these are all proteins. If we started breaking these proteins down to provide energy, well, we wouldn't even have a body for which to provide the energy anymore. So proteins are really only used for fuel when total energy or carbohydrate intake is low. That's really important to note. Right? Protein is super important, and we get that right from the popular media, but the understanding why protein is important is often confused. I see a lot of people saying you need protein for energy, but that is a false statement. We don't ever need protein for energy if we have enough carbohydrate and fat coming into the body. So, okay, we do have this term proteolysis, but yes, I guess I lied on the last page. We do have this term proteolysis. Same thing, proteo meaning protein, lysis meaning breakdown. Proteolysis is when we break proteins, especially from our diet, down into the amino acids or small peptides. But the difference I want you to get here, the reason I said we don't talk about proteolysis in metabolism is because the purpose of proteolysis is to make amino acids available for protein synthesis. Whereas glycolysis and lipolysis, those purposes are usually for producing energy. Whereas proteolysis is more so to release these amino acids into the blood, into the body for building and repair, not for energy, right? Nowhere on this page does it say, well, it does, but the primary purpose of proteolysis is to make amino acids available to build new proteins for body functions and repair. In the case that we might have excess dietary protein, right? Or in the case of starvation. But if we eat so much protein beyond what we need for building and repair, then yes, we will proteolyze, we'll break the proteins down, and we'll use the amino acids for energy, or we'll even further convert them into triglycerides for energy storage, right? So I think this is an interesting thing to note too. What this statement is saying is that if we eat more protein than we need, we will store the carbon backbones of the amino acids as fat. So remember we said we have ketogenic and we have glucogenic amino acids. Ketogenic amino acids could ultimately be converted into stored triglycerides. So we do need to revisit this process of deamination. Remember we talked about transamination, where we make one amino acid from another amino acid. That's how we can make some of our non-essential amino acids. So it's similar but different process here. Deamination is where we deaminate the amine, right? We take the amine group out of the amino acid. And so this particular image is showing a keto acid. We can also deaminate gluco acids too, or glucogenic amino acids. So deamination, where we pull the amine group out, and you might recall NH3 is ammonia, right? Ammonia, so this will happen in the liver, and then ammonia has to get converted to urea in the liver, and then urea transports to the kidney, and the kidney will flush out urea in the urine. So we have to do this because remember, glucose and triglycerides are only made up of carbon, hydrogen, and oxygen. We don't have nitrogen in glucose or in fatty acids. So we have to deaminate this carbon backbone so that we can then use the carbon backbone for energy production, right? So we deaminate the amino acid, we take the amine group off. In doing so, we produce ammonia, and then ammonia gets converted to urea, and urea gets excreted in the urine. So again, when do we see energy extracted from protein? During starvation, or if we've eaten too much, right? Those are the only two times really, if we've eaten too much protein. Those are the only two times that we see protein being used for energy. So in starvation, the body is going to turn on its own body tissues, its own enzymes, its own hormones, its own tissues, its own cells, and break those down for energy. Again, deamination, where the amine group is removed from the carbon backbone of the amino acid, again, gets converted to ammonia and then urea. The ammonia can be used, right? It can be used as a nitrogen source to make non-essential amino acids if we need to. So we might not immediately get rid of the ammonia. However, if we're not going to make non-essential amino acids right away, ammonia is quite toxic. So if we're not going to use it right away to make amino acids, then we need to convert it to urea, which is less toxic. And then again, the kidney will excrete urea in our urine. So after deamination, that carbon backbone can feed into energy production, as we saw briefly in the beginning. And it can feed into either glucogenic amino acids feeding into pyruvate, or the ketogenic amino acids feeding into acetyl-CoA, right? So again, if I go, yeah, I guess we could say it here. So the amino acids, right, that we take the carbon skeletons after we've pulled off the ammonia, after we've deaminated them. And then you have glucogenic amino acids right here, glucogenic amino acids becoming pyruvate, and then you have ketogenic amino acids right here becoming acetyl-CoA. Here is actually where we see this compound oxaloacetate. So oxaloacetate is this rate-limiting step, which is a glucose-based compound. So when I said here, going back to the fat conversation, when we say that fats burn in a carbohydrate flame, and I said that right here, there's this other compound right here, that's oxaloacetate. And oxaloacetate is made from glucose derivatives. So in order to shuttle all these acetyl-CoAs through the TCA cycle, we need enough carbohydrate effectively, or glucose substance, to make oxaloacetate. And so again, from some of the glucogenic amino acids, right, we can make pyruvate. Pyruvate can get converted back to glucose. So that's why we can make this oxaloacetate compound. And then you'll see there are some amino acids that don't fit into the glucogenic or the ketogenic groups. So there are some other amino acids that will enter the TCA cycle at these other random sites. So they can become some of those other TCA cycle metabolites. So there's no direct path, there's no singular, I guess, pathway of these carbon backbones from the amino acids. You basically have three fates. You have the glucogenic amino acids, the ketogenic amino acids, and then the others, which enter into the TCA cycle. The overview of urea synthesis again. It's not showing deamination, but deamination would happen here, right? Deamination produces ammonia. The liver can join two ammonia compounds together to make urea. Urea gets transported to the kidneys, and the kidneys excrete urea in the urine. Okay, so I know this whole protein metabolism conversation is pretty brief, as it should be, again, because our bodies primarily use glucose and fat as the energy substrate. So there's less to say about protein in this context. But obviously there's still some important pieces about extracting energy from amino acids. So please do take your time going over these. It's pretty straightforward. I know there's a lot of words here, might make it a little more confusing, but as always, email me if you have questions. So then I just want to say a couple things about alcohol oxidation. Remember on that overview slide back in the beginning, we've said that alcohol is yet a fourth macromolecule that provides energy to the body. So we do metabolize alcohol to release, to harness the energy from it. So we have actually 3 different enzymes involved in alcohol metabolism. We have something called alcohol dehydrogenase, then we have aldehyde dehydrogenase, and then we have something called the microsomal ethanol oxidizing system, or the MEOS pathway. So MEOS actually really only gets activated if you have too much alcohol. But if you were to consume like a small serving of alcohol, like 4 ounces of wine or 12 ounces of beer or even less, then primarily we're just going to use alcohol dehydrogenase and aldehyde dehydrogenase. So a small amount of alcohol can get oxidized or metabolized in the stomach, and then of course some will get absorbed out of the stomach into the blood. Alcohol dehydrogenase reduces alcohol absorption because, again, it's metabolizing the alcohol to actually provide energy. So there are genetic differences as to the amount of activity of this enzyme. So not everybody metabolizes alcohol equally well. That's why some people actually, people avoid alcohol for a variety of reasons, but some people really just don't metabolize alcohol well because they may not have a high amount of alcohol dehydrogenase activity. So they might avoid alcohol because they're not digesting it or metabolizing it efficiently. Okay, so again, we have alcohol dehydrogenase, we have aldehyde dehydrogenase. So this can happen in the stomach, and then this can happen in the liver. And ultimately what you see is we're producing acetyl-CoA. So this is still going to run into the TCA cycle. As I said, if we have too much alcohol, the alcohol dehydrogenase and the aldehyde dehydrogenase can't act fast enough. So we enter this microsomal ethanol pathway, the MEOS pathway. So it sort of subverts, it sort of bypasses ADH and creates more acetaldehyde, which then still has to be processed by aldehyde dehydrogenase. So we'll enter the MEOS pathway if we have way too much alcohol. But again, the point here is that we still metabolize alcohol down into acetyl-CoA. We're still producing energy from alcohol. So alcohol is still a source of technically calories, right? We're still making ATP. Remember, we've said a few times now, fat provides 9 calories per gram, carbohydrates and protein provide 4 calories per gram, and alcohol provides 7 calories per gram. So I guess I'll write that here. It's a random spot to write it, but fat or lipid is 9 cal per gram. So carbohydrate and protein are 4 calories per gram, and then alcohol is 7 calories per gram. So alcohol is the second most calorie dense macromolecule that we consume, right? Carbohydrates and proteins are the least calorically dense. Okay, so most alcohol is absorbed into the blood and transported to the liver to then be oxidized. So We have gastric alcohol dehydrogenase, and we also do have some of that in the liver as well. And then we have aldehyde dehydrogenase in the liver to be oxidized, again, to produce acetyl-CoA. So the liver can typically oxidize about 1 drink per hour. Again, varies on the person's genetic profile and their production of ADH and ALDH. Also will be affected by things such as overall health, the body size, right? So usually larger bodies, it's just a larger space. So they can often oxidize more alcohol at one time relative to a small body. Certain medications interfere with alcohol metabolism, and then also the nutritional status will interfere or can impede. Typically, if someone is experiencing undernutrition, if they're undernourished, then typically they're going to have a lesser capacity of alcohol metabolism. And then, of course, if there's a whole bunch of excess alcohol, that will sit around in the blood for a while until the body can metabolize it. And obviously, that's where we run into issues with overall functioning, is when we have too much alcohol in the blood. Kind of an interesting FYI or for your reference in the alcohol impairment chart. So this is showing your blood alcohol concentration for females and then for males. And in the middle here, it's showing you, safe driving limit, where impairment begins, where your driving skills become affected, such that they would even incur criminal penalties, and then your legally intoxicated penalties. So, or your legally intoxicated blood alcohol level. So you'll see it goes by body weight. And these are rough approximations. Again, it's going to depend on all of these factors here. But roughly, say, 160-pound female who's had one drink, who's had one drink is going to already be impaired. Definitely and it depends on the amount of time too. Okay, so this is 1 drink per hour. Well, no, it's not indicating necessarily 1 drink per hour, but typically your body can metabolize 1 drink per hour fine. So if you have 1 drink within an hour, you should no longer be impaired. But if somebody has, if a 160-pound female has 2 drinks in 1 hour, then she should not drive, actually. Her driving skills will be impaired. And certainly if she has 3 drinks in that 1 hour, her body has definitely not metabolized those. And you'll see by the color here, see this is that darker brown color, this is legal intoxication, having those 3 drinks in 1 hour. By comparison, a 180-pound, actually interestingly, even a 160-pound male can have 3 drinks in an hour and their driving skills will still be affected, but they don't fit that legal intoxication limit, interestingly. I think this is also helpful too. It tells you what 1 drink equals. So it's 5 ounces of wine, 12 ounces of beer, or 1.5 ounces of an 80 proof liquor. So I think, again, in this supersized food world that we live in, we're rarely pouring 12 ounces of beer anymore, or only 5 ounces of wine. So make note of that, right? Like a lot of beer cans are still 12 ounce, but a lot are now 16 ounce, right? A lot of wine glasses can hold pretty close to 12 ounces. So don't fill the wine glass all the way, fill it less than halfway. And that's 1 serving. Okay, so now we have to talk a little bit about stored energy, and this is a little review because we've talked about this already when we learned about carbohydrates and lipids. So we store energy. Hopefully you remember that we store energy as glycogen, right? Glycogen is stored glucose, but we also store adipose, right? Adipose is stored triglycerides. And we use stored energy when we're sleeping, certainly when we're fasting, and also when we're exercising. So extra energy is stored as carbohydrate. That's in limited amounts in the liver and the muscle, but fat or adipose is unlimited. That's really important to know, right? And I'm going to show you a chart in a second to give you like a numerical representation of this. So we have limited amounts of carbohydrate or glycogen storage, but we have unlimited amounts of fat. We can literally, and this is why we can become obese, is because there's no end to the amount of adipose that we can store. Again, the body has no mechanism for storing amino acids or nitrogen. So that's why we said we can convert amino acids to triglyceride. The ketogenic amino acids can be converted to triglyceride and stored as adipose, and then the glucogenic amino acids can be converted to glucose and potentially stored as glycogen. So this is kind of a cool chart. So for a well-fed 70-kilogram male, that'd be like 154 pounds. So, okay, so for a 154-pound male, they can store about 800 calories of glycogen, but they can store about 135,000 calories of triglyceride, right? And that's for 154 pounds. So if this person was, 250 or 350, there's going to be a major increase in the triglyceride. The amount of glycogen that this person can store won't change very much, even as their weight goes up, because really when we gain weight, we're mostly gaining adipose tissue. You'll see over here too that we store quite a bit of protein, but we're not storing it. We're not actually storing amino acids. We are made of amino acids. Our bodies are made up of proteins. Again, this is like any organ in your body is made of proteins. Transport molecules in your blood. Your blood is made of proteins. Your immune system is made up of protein. And the enzymes that do work are made of protein. So this isn't stored energy as protein. It's just saying that if you were to utilize your body as an energy source, you would get about 2,400 calories, 24,000 calories of energy from your protein. So again, we don't store a lot of glycogen, which is just kind of an interesting to note, but we really have endless reserves of triglyceride. Okay, so really so far we've been talking about catabolism. So now let's review a little bit of anabolism that we've talked about already. So remember this process of gluconeogenesis, making new glucose from non-carbohydrate precursors. This is always really important. This is another question that I ask folks, or I'll ask you on your quizzes and exam for Unit 2. Gluconeogenesis necessarily makes glucose from non-glucose. So if it's not glucose, it's going to be amino acids or it's going to be triglyceride, right? So we make gluconeogenesis makes glucose from glucogenic amino acids and from glycerol. Remember, fatty acids can't make glucose, but the glycerol can. The glycerol can get converted to pyruvate, and pyruvate can get converted to glucose. So where do we see gluconeogenesis occurring? During sleep, during fasting, potentially during trauma, like a physical injury, and during exercise. So protein catabolism for glucose production can draw on vital tissue proteins like skeletal and heart muscles and various organs, right? This is why we don't want to see protein catabolism. This is why we want to eat enough. Carbohydrate so that we don't need to start breaking down our own body proteins to be used for energy. This is also something that can happen in a poorly managed ketogenic diet, is that you can actually just start breaking down your own body proteins, you can actually start breaking down your own body proteins to be part of this gluconeogenic process. When somebody is entering this or trying to enter ketosis, these processes of gluconeogenesis are going on, right? Because you're not providing enough glucose to the body. This is a long lecture, I guess, so my computer isn't using its willpower. Alrighty, so gluconeogenic processes, right? Proteins So gluconeogenesis, we're going to make glucose from either triglyceride or from proteins. Again, it's really only specifically glucogenic amino acids that make glucose and glycerol that can make glucose. We also have lipogenesis, again, lipolipid genesis creation, de novo synthesis, so again, new synthesis. So it's the production of fat from non-fat substances. That's what de novo means. It's making fat from non-fat substances. Well, what are the non-fat substances from which we make fat? Carbohydrates, ketogenic amino acids, and alcohol, right? Hopefully I have a chart there, yeah. Right. Carbohydrates, we can make pyruvate, which can make acetyl-CoA, and from acetyl-CoA, we can do the reverse of beta oxidation, and we can build fatty acids from acetyl-CoA. So where else did we make acetyl-CoA? We made acetyl-CoA from ketogenic amino acids, and we made acetyl-CoA from alcohol. So any of these, any excess of glucose, any excess of alcohol, any excess of ketogenic amino acids can lead to lipogenesis or fat buildup, fat creation. So this happens when we consume extra calories. Yeah, and this process takes place in the liver. Lipogenesis, creating fat synthesis from non-fat substances. And then we can also make proteins, right? Amino acid synthesis. So the body can make the non-essential amino acids, right, through that process of transamination. And this can only happen when we have enough energy and enough nitrogen. And remember, essential amino acids must be consumed in our diet. So enough energy, meaning we've consumed enough calories, we've taken in enough food, and enough nitrogen, meaning we have also consumed enough protein so that we have a robust amino acid pool, which will provide that nitrogen in the amine group for this process of transamination. And just a review of transamination and making non-essential amino acids. So amino acid A, right? This is the side chain. Where are we going here? This chart always confuses me. So in transamination, you're going to transfer, right, your amine group. So you start with some sort of, I guess down here, you start with some sort of backbone, but then you need the amino acid group added to it to become, you need the amine group added to it to become an amino acid. Same here, so we've got the keto acid backbone, but it's missing the amine group. So when you add the amine group to it, this keto acid becomes the amino acid. Alrighty, and then we'll review some of the hormones that regulate metabolism. So we have insulin, glucagon, epinephrine, and cortisol. Remember, we talked about all of these in managing our blood glucose. So insulin is anabolic, right? Meaning it's going to support synthesis. So we see insulin after a meal. It activates storage enzymes. So insulin's job is to encourage uptake of glucose, but it also has some other jobs. It also encourages cells to take up fatty acids and amino acids. So insulin regulates not just glucose absorption, it also, not absorption, but glucose uptake by the cells. It also supports or promotes fatty acid and amino acid uptake by cells. Whereas these other three, glucagon, epinephrine, and cortisol, are all catabolic. They support the breakdown or signify or initiate the breakdown of stored triglycerides, of stored glycogen, and stored body protein. So we break any of these things down to start to provide energy. And if you go back to the carbohydrate chapter, chapter 4, remember we talked about all of these, and we talked about all of these different processes of building glycogen, right, or breaking down glycogen, or breaking down stored triglyceride, lipolysis, right, breaking down stored triglyceride, breaking down body proteins. So it might be helpful to also go back and review chapter 4 with these hormones. This is also a helpful chart. So you've got the hormones listed here, and then you'll see the metabolic state. So fed, fasted, exercise, or stress. So remember glucagon, epinephrine, and cortisol. So when we're fasted, when we're exercising, or when we're under stress, this is all catabolic, right? Whereas insulin is when we're fed, right? We're well fed, we just received a bunch of food, so now we can build, right? Now we can do synthesis. So let's see. I don't think I'm going to spend a lot of time on this chart because we have talked about all of these things already. So do take a little bit of time to go through and, like, if you haven't really remembered much of this already, try to remember how each of these 4 hormones affect carbohydrate metabolism, lipid metabolism, and protein metabolism. Again, pay attention to whether they're catabolic or anabolic, because that's going to help you recognize whether it's synthesis or whether it's degradation. Remember, gluconeogenesis, even though it's a synthesis reaction, it's also necessarily a degradation of the substances that provide the substrate to make glucose. So gluconeogenesis is a degradation of protein. Gluconeogenesis is, which is what you see over here, degradation of protein. Gluconeogenesis is a degradation of fat, lipolysis, right? So it should be pretty straightforward. Okay, so let's talk about what the anabolic state means. So again, this is after we've eaten a meal in response to feeding. So we're in a fed state, meaning we're fed, we've eaten, we have nutrients, we have calories in the body. So the bloodstream is then going to be enriched with all these good nutrients from the food, glucose, fatty acids, and amino acids. So what are we going to do then? We're going to build. So we're going to build glycogen. We're going to build adipose, and we're going to build new proteins, or as needed, we will deaminate amino acids so that we can convert the carbon skeletons to fatty acids and store them as triglycerides. Same thing with glucose. Once we've stored enough glycogen, we will convert glucose to acetyl-CoA and then build fatty acid chains and store triglyceride. Again, also a helpful chart here to look over the fed state. So it's generally anabolic. After digestion, absorption, and transportation, the end products of digestion will be synthesized into important biological compounds or used for energy or stored as energy, right? So amino acids will be used to synthesize body proteins and muscles. Excess amino acids can be used for energy or converted to fatty acids, which will be stored as triglycerides. Carbohydrates, we render glucose, that's primarily for fuel. Excess glucose will be stored as glycogen in the liver and the muscle. And once we max out our glycogen stores, right, we have very limited amount of glycogen store, then glucose can be converted to glycerol and to fatty acids to also build your adipose. And then the same thing with triglycerides, right? We break those down to make fats and glycerol. These can provide fuel, and again, excess will be stored as adipose. So you'll see also up top here that can be helpful. Red is indicating catabolic reactions, blue is indicating the synthesis or the anabolic reactions. So again, we kind of talk about them separately, but it's also hard to talk about catabolism and anabolism separately because they just interweave so tightly. And so again, all of this is metabolism, right? The sum of all the processes of catabolism and anabolism is what metabolism is. Okay, so then we've just talked about the fed state, so now let's talk about the fasting state. So this is short-term fasting, so this might be, 24, 48 hours. So short-term fasting is going to stimulate catabolism. That should make sense, right? If you don't have energy coming into the body, we've got to go find it. So we're going to break down glycogen, release glucose into the blood. We're going to break down stored adipose as well. So we're going to see a lot more fatty acid in the blood. Fatty acids will get converted to glucose. Maybe not quite yet, I guess. Glucose synthesis from glucogenic amino acids and glycerol. So sorry, I didn't mean to say fatty acids. Fatty acids will undergo beta oxidation, right? Because fatty acids cannot make glucose. Only glycerol can make glucose. So what little glucose remains will be conserved for the brain and other cells, like your red blood cells, that can only rely on glucose as fuel. So ketones form as acetyl-CoA units are blocked from entering the TCA cycle. So remember, and when we talked about fatty acid metabolism, fatty acid catabolism, we said that we get these buildup of ketone bodies. This happens primarily when there isn't enough glucose coming in because that oxaloacetate molecule isn't built. We don't have enough oxaloacetate for these acetyl-CoA molecules to run through the TCA cycle. So that's primarily where the ketone bodies get built up. But then those ketone bodies can be metabolized themselves for energy. So if fasting were to be prolonged, like really over a series of days, that's when we enter into starvation. So the body shifts into survival mode. So blood glucose is still maintained to support the brain and the red blood cells. Fatty acids will become the primary fuel. The brain will begin to use ketone bodies for fuel. And we'll start to break down body supplies. So we'll break down muscle tissue, we'll break down heart tissue, we'll break down other organs and convert those proteins into glucose as much as possible or into fatty acids to supply energy. And so we're going to see a decline in overall activity. We're going to see body temperature go down. We're going to see overall metabolism go down. So the body's just going to slow down and try to conserve as long as it can. But obviously in starvation, we're not getting new calories. We're not getting new energy into the body. So if that persists, then ultimately death results. So for as long as the body can, it's going to use its adipose tissue and it's going to start to break down its body proteins to provide energy. Remember back on that chart, we said that a 150-pound male has like 24,000 calories from the body proteins, right? So that would last a little while, but ultimately you can't break down your whole body and still be alive, right? So yes, you can supply some energy by breaking down some of your organs, but ultimately death would then result. Okay, and then a similar chart, but just looking at metabolic responses to fasting. So the other chart was in the fed state. This is now a fasting state. So fasting is generally catabolic. We're going to see a lot of breakdown. So after a period of fasting, when the body glycogen stores are reduced, the body is going to increase its use of stored fatty acids. So muscle glycogen and liver glycogen are going to get broken down in short-term fasting, right? And they're going to provide fuel. Remember, we said there's only about 800 calories of stored glycogen in a 150-pound male. So 800 calories probably won't get us through even one day. We're also going to be breaking down adipose and using the glycerol and the fatty acids for energy. So then as short-term fasting becomes long-term fasting, we start to border on starvation here, right? So we're going to start to break down muscle. We're going to start to break down other body tissue. So the amino acids, and we're going to break down triglyceride. We're going to break down adipose. So the glycerol and the amino acids will be sent to the liver to be converted to glucose. And then glucose will be primarily reserved for the brain and the red blood cells because they can't really function on ketone bodies. And then eventually we're going to see the liver will make ketone bodies from fatty acids. Fatty acids, remember, can't be converted into glucose. So eventually body cells are going to have to start using ketone bodies as well as, well, we've already been breaking down amino acids, right? So some of these amino acids are going to contribute to the ketone bodies, the ketogenic ones are. So then we're still relying really on glucose and ketone bodies, but the sources of glucose become your stored body proteins and your stored adipose. Okay, so that was a really long lecture. Definitely email me with questions. Definitely, I know if you're at the end of it already, go through it in chunks. But if you can, take some time and go back and review. I'll go through these review questions here, so you can also take some time and test yourself on these. Yeah. Alrighty, so again, thank you for listening. Email me with questions. Take your time with this material. This is the only lecture for this week. I put it all into one lecture for chapter 7. So go through it once, go through it twice, but definitely take whatever time you need to really solidify these concepts in your brain. Alrighty, thanks everybody, and we will see you next